Okay, so thank you very much and welcome everybody for this Global Virus Network Forefront of Virology webinar. And as always, if some of the attendees do not exactly know what the GVN is about, I will make a very short introduction. Global Virus Network has been created in 2011 by Bob Gallo, Billy Hall, and at that time, Reinhard Kurt. And this is now, it includes now about 70 research centers worldwide on all continents and 12 affiliate centers. And this is, the keywords are science-driven and independent, with 3 main activities: research with the task force, with the watch groups, and you are always most welcome, and also for the other GVN members, to go on our website and maybe be included in some of the ongoing activities. Second, education training with the GVN Academy, a lot of focus on this. Again, you find all the details on the website. And finally, advocacy communication. And of course, these webinars are part of the scientific activities and communication. So it's really in this context my great pleasure to welcome Dr. Michaela Gack. Michaela has been trained in Germany at the University of Erlangen-Nuremberg. Then she had her Ph.D. in a joint effort between this university and Harvard, then moved to a postdoc in Los Angeles in the University of Southern California, then back to Boston at Harvard, then in Chicago, where she became a full professor, and finally at Cleveland, and she is presently the scientific director of the Cleveland Clinic Florida Research and Innovation Center in Port St. Lucie, thanks to the Arthur and Marylin Levitt Endowed Chair. And she's also a professor of molecular medicine at the Cleveland Clinic Lerner College of Medicine of Case Western Reserve University. So Michaela has a very, very interesting career track and a lot of achievements, focusing on the interplay between viruses and the host innate immune response, and also on the characterization of host enzymes, which are very important for the virus life cycle. And in fact, today, she is going to discuss some of her results. Before closing, I really want to thank Bob Silverman. Bob Silverman has been the director of the Cleveland GVN Center, always very committed. He has retired, but we know that he will still be very interested in the GVN. And Michaela is now the director of his GVN Center. So again, a great pleasure to welcome you, Michaela, and we look forward to your lecture. Thank you.
It's my pleasure to tell you about, our research on innate and intrinsic host enzymes. Today, I will be mainly talking about host enzymes that modify actually the virus and thereby regulate essential enzymes in the virus life cycle. But first, a little bit of introduction. So can you see, I'm sorry to interrupt one more time. Can you see the title well, or?
Yeah.
Yes, okay, perfect. So over the past century, we have seen, a number of either newly emerging or re-emerging viral pathogens, mostly RNA viruses, and some of them are illustrated here. Most of them has caused outbreaks, and some of them have also caused quite devastating viral pandemics. And listed here in the numbers are the death tolls that each either outbreak or pandemic had. Just to mention a few, there was, of course, the the Spanish flu 1918, when it started, that caused an estimated of 40 to 50 million deaths globally. The still ongoing AIDS pandemic, currently it's around 40 million, the death toll. The swine flu from 2009, and then of course COVID-19, the current numbers are around 6.9, close to 7 million deaths worldwide. When you see all these different, pathogens that have caused outbreaks or viral pandemics, I think it is fair to say that there are only few antivirals available. Of course, there are some success stories, one of which is for HIV. There are many, antivirals available, but all taken together, again, I think it's fair to say that we have relatively few effective antivirals. And for some viruses, we don't have any specific antiviral. The current paradigm of antiviral development is to directly target the virus, in most cases, targeting viral enzymes, but also, preventing entry, for example, of viruses, and there are other, mechanisms. The current paradigm of antiviral development is also post-emergence, so development after the virus emerged, and it is typically single virus targeting. So it only works for one specific virus and not necessarily for other viruses, for example, in the same family. So there are certainly some caveats of this approach, one of which I already mentioned. It means that it's single virus targeting. So typically these antivirals that target directly the virus are ineffective against new viruses. And all of you, know that over time, these approaches can, cause drug-resistant viral strains, mainly because the virus is mutating and thereby evading, these antiviral compounds. So a complementary approach in antiviral therapy has been the host-directed antiviral therapy. It is expected to be more broadly acting than maybe the virus-targeting one. It could potentially avoid viral resistance, and generally it only works if host cell functions and integrity are maintained. And principally, you can group these host-directed antiviral therapies into two main groups. The one is to elicit either directly or to boost an antiviral immune response. One example is, for example, the interferon alpha therapy for hepatitis C virus or other chronic infections. But then the other category is to target host cell factors that are essential for virus replication. One example here is CCR5 antagonists for HIV-1. In recent years, we have really seen fantastic, approaches and new methodologies to get to these host-directed antiviral therapies. And really the first step is to identify these key factors inside cells that could be, potentially be blocked as a host-based antiviral therapy. So key in these approaches are certainly, different proteomics approaches, classical affinity protein mass spectrometry, global proteomics approaches, but also systems biology, pathway enrichment, kinase predictions. This all has really helped to identify essential host factors, driving or allowing virus infection and replication. And not to forget gene targeting screens such as CRISPR knockout screens that also have been really instrumental in identifying these host dependency factors for viruses. Today, actually, I want to talk about one small subset of these host factors that could be potentially promising targets for host-based antiviral therapy. And these are human enzymes or host enzymes that post-translationally modify viral proteins. Maybe a little bit of background about that. We all know there are many different post-translational modifications. They can be very small chemical groups such as phosphorylation, acetylation, or methylation, to very large, proteins and even polypeptides such as ubiquitination that can be attached to the target protein. And we know mostly PTMs, how we call them, mostly for, cellular processes. Essentially, post-translational modifications, they regulate any single step in a cell from signal transduction, epigenetics, metabolism, and immune responses. And in most cases, these post-translational modifications, they can change structures, meaning inducing structural or conformational changes. They can modulate the interactome of a protein or change its localization of a given protein. And as you know, they are reversible typically. So there are, enzymes which catalyze it and enzymes which remove these specific PTMs. Most well known, of course, kinases versus phosphatases, ubiquitin E3 ligases and DOBs, and acetyltransferases and deacetylases. My lab over the past several years has mainly studied, innate immunity, innate sensing mechanisms by these different sensors, toll-like receptors. We mainly focus, I should say, on RIG-I-like receptors, but you can see there are multiple different sensors, other ones, C-gas and NOD-like receptors. And we mainly studied how post-translational modifications regulate these early steps in virus sensing and the ensuing type 1 or type 3 interferon response. And listed here in green and red are all kinds of modifications that have been identified to regulate either directly these sensors, such as ubiquitination and acetylation, phosphorylation, and many more, or that act somewhere downstream in the pathway to regulate an antiviral response. So this is only one, aspect how PTMs regulate, antiviral responses by, for example, regulating innate immunity and type 1 interferon response. But overall, I think it is fair to say as compared to the human PTOM, so what we know about PTMs in on human proteins, our current knowledge about the viral PTOM is really still rudimentary. And today, as an example, I would like to present some recent data from my lab that were published this month in August on how flavivirus replication is regulated by the host acetylation machinery. We all know, acetylation most well. We know it from epigenetics because nuclear residing acetyltransferases, we know they can regulate and acetylase histones, and this will regulate, chromatin formation and thereby epigenetics. But we found a very different mechanism in flaviviruses. We found that a host acetyltransferase directly acetylates the helicase of these viruses and thereby directly regulates functions, essential functions of the helicase. So it's a very different concept how acetylation can regulate a virus infection. A little bit of background and information about the flaviviruses. As many of you know, they are really, have a huge, health burden globally. Most of them are transmitted by insects, such as mosquitoes or ticks, and can be also other insects. I highlight here on the top dengue virus that alone, causes at least 390 million infections, and nowadays actually even more globally. So a huge health burden, just one single virus, dengue virus with the 4 serotypes. But then also Zika virus is here highlighted, yellow fever virus. And then also you can see also the widespread distribution of West Nile virus and maybe a little bit more restricted, but still, huge health burden, Japanese encephalitis virus. And then there's this whole group of, I call them now, more recently emerged or understudied flaviviruses, such as Powassan, Usutu, Spondwani viruses, and more, that also cause a potential risk, for spread and also on human health. These flaviviruses cause a wide range of diseases, including neurotropic diseases, encephalitis, for example, in case of West Nile virus, but also visceral diseases, hepatitis, and vascular leakage and hemorrhagy and bleeding. And then in some cases, such as Zika virus, they can cause congenital disease. We all, have heard about, the microcephaly, for example, placental insufficiency. So there is really a wide variety of diseases caused by flaviviruses. In my lab, and this is really the topic of the talk today, we were interested in one key enzyme of flaviviruses that all flaviviruses have is the NS3 protein. The NS3 protein is really an interesting enzyme. It has actually 2 enzymatic activities. It has a serine protease at the N-terminus, and this is involved in viral protein, polyprotein cleavage. So flaviviruses, when they enter the cell, their positive sense single-stranded RNA genome is directly translated into one very large polyprotein. And this is then subsequently cleaved into the individual proteins, 3 structural ones, and 7 non-structural ones. The non-structural ones then form mainly the replication complex at the ER to replicate the viral RNA. So NS3 participates in this cleavage of the polyprotein, so it's really an essential protein in the virus life cycle, again, absolutely needed for virus replication. The second enzymatic activity is a helicase function at the C-terminus, and this is really here acting in the replication complex where it unwinds double-stranded RNA intermediates and thereby allows for efficient viral RNA replication. Because of these two absolutely essential enzymatic activities, NS3 has been a major target for antiviral drug development for these flaviviruses. And it has been still challenging, I mean, to find, effective and very effective drugs targeting these enzymatic activities. So conceptually, we wanted to take a different path and ask the following question. Can we identify novel host regulatory mechanisms of NS3 enzymatic activities, which may spur new antivirals targeting strategies? And specifically, we wanted to identify post-translation modifications in this protein NS3, and maybe thereby providing insights into how they could maybe regulate enzymatic activities. And what we found and what I will be presenting today is our new study on NS3 acetylation. And this is we found by 2 complementary mass spec approaches. On the one hand, we purified overexpressed, just expressed from a plasmid NS3 from Zika virus. I should mention we started out with Zika virus. I will be later showing you data also on other flaviviruses, but the whole study started with Zika virus as one of the significant flaviviruses. So we either overexpressed just the NS3 protein in cells. In this case, we also treated with interferon to kind of mimic an infection state for our mass spec. And the second approach was we used authentic Zika infection in astrocytic cells, SVGS cells, and then purified the whole viral acetylome after enrichment with an acetyl-lysine antibody. And again, we subjected these samples to mass spectrometry, and this identified the helicase domain especially as a major target for acetylation, because we found 5 residues in the helicase domain. 1 we also found in the protease domain, as shown here. And just for clarification, the red indicated lysins we found actually in both mass spec approaches, whereas the black ones here, highlighted ones, we found only in the overexpression. To really find out, which of these lysins, these acetylated lysins, are the dominant acetylation sites and the functionally relevant ones, we mutated each of these 6 lysins to arginine, which cannot be acetylated, and then by using biochemistry and an antibody, it's a pan-acetyl-lysine antibody, we compared the acetylation of these mutants compared to wild-type NS3 of Zika. And what we saw is that there's one lysine 389, that when we mutated it, we got a marked reduction of the acetylation, which suggested to us that this is the dominant or major acetylation site in Zika NS3. We also can readily see this acetylation of NS3 during infection. These are again human astrocytes, which we infected with a Brazilian 2015 strain, and this is what we use in most of our experiments, Zika virus. And we see after we immunoprecipitate NS3 from these infected cells, we see an increase of acetylation over time, which also correlates with the increase of NS3, which is a non-structural protein and increasingly produced during infection. We also generated an acetyl-lysine 389 NS3 Zika antibody that allows us to specifically monitor this acetylated lysine of Zika. I should say we have validated the virus, validated the antibody, and it works pretty well to monitor acetylation of Zika NS3 during infection at this specific lysine 389. But what was very interesting to us when we looked into the structure of NS3 helicase bound to a synthetic mRNA, and this structure had been already solved exactly in 2016, we found that this lysine lies within the RNA binding tunnel of the NS3 helicase. And this was very interesting to us because of the following reason. From epigenetic and histone modification, we know that acetylation can regulate the charge at the lysine. Lysines are typically positively charged. The acetylation neutralizes the charge. In the case of histones, modulates the DNA binding ability and thereby chromatin formation. So we wondered if, this acetylation and potentially changing the charge at this lysine here can also regulate RNA binding and other functions of the helicase, kind of similar to what's happening with histones and DNA binding. So to do so, we performed an in vitro RNA binding assay using a biotinylated Zika RNA that was synthesized. And we generated multiple or 2 mutants for Zika NS3. One of which I already mentioned is the arginine mutant. So arginine, it cannot be acetylated, but it still keeps the positive charge. And the second mutant is glutamin, which will neutralize the charge. And in many cases, it has been shown in other studies for other proteins, it can mimic an hyperacetylated state. So we subjected these mutants, these key mutants of NS3 as compared to wild-type NS3 in this RNA binding in vitro assay. And we observed that this R mutant had a higher binding ability as compared to wild-type, whereas the Q mutant had a strongly diminished RNA binding ability. When we tested, however, unwinding activity using a double-stranded RNA that can be unwound in this helicase assay, we saw that this Q mutant practically had abolished or almost abolished, unwinding activity. It's strongly reduced, but also this R mutant, which actually had a higher RNA binding ability, also had a reduced unwinding activity. So we concluded from these data that locking, NS3, even in the very robustly RNA bound state, so kind of a rigid RNA binding, still is not optimal for the unbinding activity. But as it relates to acetylation, we think there is a dynamic and reversible acetylation needed to really, have RNA binding and unbinding, especially activity, that you need this dynamic and reversible acetylation at the site for optimal unwinding activity. Just to mention it, I'm not showing here the data because of time, but we have tested this mutation also on other critical activities of NS3. One of it is protease activity together with NSO2B. It had no effect on protease activity, not on NS5 binding. We also tested one of the immune evasion mechanisms of NS3, RIG-I signal inhibition. And just to state this, we did not see any effect of mutating this lysine on these other activities. Next, we generated fully infectious clones of Zika virus, so recombinant Zika virus. And we, again, introduced this arginine at lysine 389, so mutating it to arginine or to glutamin, and then compared these mutant virus in their replication capacity as compared to the parental virus. And we observed, similar to our helicase assay, which showed, reduced activity, also that both viruses were attenuated, here shown by Zika E transcripts over a time course in astrocytic cells. And also here shown by titers. The Q mutant is quite, strikingly attenuated, but also the R mutant, again, correlating with our results in the helicase assay. I should say we have seen similar attenuations in other cell types. Here I'm showing you astrocytic cells. We also have tested microglia cells, more recently also, different kinds of cell lines, including A519 viro cells. So we see in the cell lines that we have tested so far a similar attenuation phenotype for these mutant viruses. Interestingly, when we passage these mutant viruses over time, they reverted back to the wild-type codon by passage 3, which correlates with a restoration of virus growth. And we believe that the virus really needs this lysine, which can be reversibly acetylated, rather, again, locking it in one of these states either R and Q, meaning non-acetylated or acetylation mimic in this case, is not optimal for the virus, as also our helicase, data indicated. So we concluded from this that lysine 389 acetylation in Zika NS3 is a proviral regulatory mechanism through which the virus achieves efficient RNA unwinding and ultimately viral RNA replication. So we wanted to identify the enzyme which catalyzes this NS3 acetylation. And, flaviviruses are very small with having 10 proteins. So we knew it must be a host acetyltransferase which catalyzes this critical event in NS3. So we undertook an RNAi screen silencing all human acetyltransferases, and there are not too many, luckily, as compared to kinases. So the human acetyltransferases can be subcategorized into canonical, non-canonical, and putative acetyltransferases. And you can see we silenced individually all of them or each of them as compared to control as iRNA. And we tested first their knockdown, the effect of the knockdown on Zika replication by measuring Zika E transcripts using qPCR. And you can see that it's actually exactly 9 acetyltransferases. When we silence them, Zika replication is markedly or significantly reduced. The 9 so-called, how I call it, proviral candidates are listed here. Just to mention it, even though this was not the focus of our study, we also found several acetyltransferases that when we silence them, actually Zika virus replication is enhanced, so they could be potentially antiviral acetyltransferases. But of course, with these 9 proviral candidates in hand, we couldn't tell, where exactly they act in the flavivirus life cycle. They could act, on many different steps, either on the viral side or also on the host side. So we subjected as a next step these 9 candidate acetyltransferases in a biochemical assay, where we again individually silenced them, but now directly monitored the Zika NS3 acetylation exactly at this residue lysine 389. And what we saw in this biochemical assay, that only silencing of one acetyltransferase, Cat5, markedly reduced acetylation of NS3, whereas the other ones did not have any diminishing effect on NS3 acetylation. We also readily found that Cat5, this acetyltransferase bound to NS3, this is again during infection inside cells. We readily can see by co-IP study this interaction. And we also wanted to compare the effect of silencing this newly identified, what we show proviral factor Cat5, this acetyltransferase, to other known host dependency factors of flaviviruses. One prominent one is TMEM41B. That's an essential host dependency factor for Zika virus as well as for other flaviviruses. And we could observe when we silenced Cat5, we got a tremendous reduction of Zika transcripts, and either equally or even higher than TMEM41B, shown here in A519 cells, but we have same data or similar data from astrocytic cells, microglia cells. Most recently, we have tested neurons, and we see in many different also cell lines, viral cells, also a strong reduction of Zika replication when we silence Cat5. A little bit more information about Cat5, and Cat5 comes actually in 4 flavors, meaning 4 different isoforms in humans, which are listed here. So they are called Cat5L for long, alpha, gamma, and beta, and they distinguish themselves by the inclusion or splicing of either intron 1, here shown in blue, or exon 5. And when you look at a whole cell acid and you probe with a pan-Cat5 antibody, you actually nicely can see these different isoforms if you separate it well enough in a gel from the long, alpha, gamma, and beta. From all these isoforms, actually, if you look into the literature, mainly one of them only, Cat5 alpha, which is most well known under the name TIP60, is well characterized functionally. And it is an epigenetic regulator. Cat5 alpha, some people don't call the alpha, but it's actually alpha what they study. It's nuclear residing in most cells and thereby regulates, epigenetics and histone modification among some other functions. Why do I tell you about these isoforms? So when I showed you this CoRP study already, when we performed interaction studies with Zika NS3, we only saw one single band being bound to NS3. We knew it is at the height of gamma, the isoform. And just for comparison, for you to show, if you look at the whole cell lysate here, you see again nicely these 4 isoforms. But again, when we do binding studies, only one single form or band bound to NS3, which corresponded in size to the gamma isoform. But we really wanted to corroborate that if it is indeed the gamma isoform that mediates this NS3 acetylation event. So we undertook a specific silencing approach where we designed siRNAs targeting intron 1, which will then silence L and gamma, as well as one which targets exon 5 and thereby silences L and alpha. And as a control, we also designed siRNA where we silence all, which we actually used before in our other experiments, the data what I showed you. And this silencing approach really works. You can see, L and gamma is here silenced, and here L and alpha, the upper 2 ones, and then the all siRNA will silence all of them. So it works actually very well. And then we tested the silencing, pairwise silencing on NS3 acetylation, shown here left, but also Zika replication. And what we saw was when we silence L and gamma together, we see a marked reduction in NS3 acetylation at 389, but not when we silence L and alpha, indicating it's the gamma. We also see a reduction when we silence all of them. And the same is actually here reflected in Zika e-transcripts as a measure of Zika replication. When we silence L and gamma, we see a strong reduction of Zika transcripts, similar to when we silence all Cat5 isoforms. But when we silence L and alpha together, there is no diminishment of Zika replication. Again, all indicating that is the gamma isoform, which I can tell you has never been studied functionally at all, that this is the one mediating Zika NS3 acetylation. We also showed this in a different way, whereas wild-type Zika Cat5 gamma can strongly, enhance NS3 acetylation. We saw when we created a dominant negative mutant of Cat5 gamma, it diminished Zika NS3 acetylation. And when we transfected increasing amounts of this dominant negative mutant of Cat5 gamma into cells, we actually can robustly decrease Zika virus replication, here shown by Zika positive cells and by flow cytometry analyzed. What is the effect of Cat5 gamma silencing on other viruses? Again, we are not able so far only targeting, unfortunately, the gamma, so it's silencing for L and gamma, just to clarify that again. But we didn't see any effect on, for example, EMCV replication as a picornavirus family member, or VSV, vesicular stomatitis virus, which is a rhabdovirus, whereas the effect of Zika is really striking. These are viral titers, what are shown here. But then we also wanted to know more about, so how does this work? What is so unique about this gamma isoform, again, that has never been studied in any context functionally? So we first thought maybe, this is an inducible isoform, maybe, transcriptionally inducible, but we did not see that. So in pink here shown total actually Cat5 transcripts, we don't see any upregulation of Cat5 during Zika virus infection. In gray is just the control that's actually measuring the E transcript of Zika just as a control of infection. And again, here shown in A509 and here in the astrocytic SVGA cells, Cat5, none of the isoforms we think we see upregulated transcriptionally. But also, even though I don't show here the plots, I maybe briefly showed it earlier, we don't see any increase also on protein level on any of these isoforms upon Zika infection. So we wanted to know, so what else could it be? What makes this gamma isoform so unique and allowing, NS3 acetylation? I should mention, acetyltransferases and also Cat5, the other isoforms, especially alpha, are mainly known to reside in the nucleus because they regulate, epigenetics and gene transcription. But NS3 of Zika virus and flaviviruses in general, they are maturely localized in the replication complex at the ER. So we wondered if there's maybe some kind of relocalization or recruitment of Cat5 gamma to the viral replication complex. And to study Cat5 gamma in isolation, again, there are these 4 isoforms, which makes everything a little bit more complex. We constructed Cat5 gamma reconstituted cells. So what that means, we stably silenced all endogenous isoforms and then put back Cat5 gamma alone, put it back, allowing us to monitor only this specific isoform during Zika infection. What we saw is, and Cat5 gamma is here shown in green in these reconstituted cells, in an uninfected cell, it still resides in the nucleus, what you can see overlay with a DAPI staining. But when we infected cells with Zika virus, and these are here 2 different MOI, 0.1, and then we increased it by 10-fold to MOI of 1, we saw that there is a relocalization, apparently, or I say now redistribution from Cat5 gamma going out from the nucleus and localizing with NS3, here stained in red, and co-localizing it here. We have also stained, with ER markers. We know this is the replication complex. And this, redistribution is really striking. If you go to an MOI of 1, you see really a striking redistribution of Cat5 gamma, co-localizing then at the ER, what we know at the replication complex. We have done that with multiple different markers for ER. Here I'm showing you NS4B. That's another flavivirus protein that is at the replication complex. And here again, these are uninfected cells where Cat5 gamma is primarily localized to the nucleus, but you see this redistribution upon Zika infection and co-localization with NS4B as well. The other question what we had is NS3 acetylation by Cat5 gamma conserved among the flaviviruses? And I can tell you the answer is yes, at least for the members we have tested so far. I'm showing you only a few data because of time, but we could readily see acetylation of NS3 in infected cells, authentically infected cells, for West Nile virus, for dengue virus, that's a serotype 2, and for yellow fever virus. In this case, we used a different approach. We used IP enrichment for acetylysin and then probed with NS3. So NS3 is a robust acetylation substrate, again, during authentic infection. And even though I'm not showing you all the data, but we could confirm that it is Cat5 gamma mediating this acetylation event also in West Nile virus, dengue, and yellow fever virus. These are the members so far that we have tested among the flaviviruses. And also in these cases, we find that Cat5 gamma is a conserved, we think, proviral host factor, again, not only for Zika, but also for West Nile, dengue, and yellow fever virus. So again, this is the pairwise silencing approach where we see that for West Nile virus, I'm showing you here titers, for the other flaviviruses transcripts, but in either case, we see that silencing, whenever you silence gamma, you see a reduction strong either in titers or transcripts, but not if you silence Erlangen alpha, or alpha, I should say, or you don't see any significant, maybe a minor reduction, but not a striking reduction. And the same is here also for yellow fever virus, measuring transcripts. Again, reduction whenever you silence gamma. So I think I would like to conclude, here and tell you, what we learned from that and what maybe also are the next steps. But we found that Zika virus NS3, the helicase, is a major target for acetylation. And to our best knowledge, this is the first known example that a viral helicase can be acetylated, and that regulates the helicase activity. and thereby virus replication. We have identified as the dominant residue 389, which lies in the RNA binding tunnel. We also identified what I showed you, that Cat5, but only one specific isoform, the gamma isoform, mediates this event. And it appears that it gets relocalized outside from the nucleus onto, where the virus replicates here on ER membranes in the replication complex. We show that Cat5 gamma is a proviral factor, really mediating and supporting not only Zika virus, but multiple different flaviviruses, and we are testing now additional flaviviruses. And we suggest that Cat5 gamma may be a potential target for pan-anti-flaviviral therapy. Of course, that would need to be tested, thoroughly. But conceptually, I want to put out this question, what we are here in the lab addressing. Can we block essential viral enzymes indirectly by targeting their host modifying enzyme, these PTMases? Such as here the acetyltransferase, obviously they are kinases which could modify, different essential viral enzymes and many more, enzymes. So this would be an indirect approach to target still these essential enzymes, but again, indirectly, not how we traditionally do it, directly targeting them. Last but not least, and actually most importantly, I would like to thank all people, who did the work. And I can tell you it was really a group effort here, but it was led, and also the original discovery and many of the data were from Taryn Sherman in the lab. Also, I would like to note Cindy Chiang, who significantly contributed, as well as several other people in the lab, Kirosh Acharya, Hejong Lee, Liu Liu, Santoshi Mopala, Shanti Pandey, and also Suba Sayad, who contributed to this work. And importantly, also our collaborators, Konstantin Sparrer and Meta Wolczyk at the Ulm University in Germany, and Sean Stauffer and Christopher Goins at Cleveland Clinic in Ohio. And with this, I would like to end, and I'm happy to take, any questions. Thank you.
Thank you so much, Michaela. Great talk, and a lot of very, very stimulating findings and hypotheses, exactly in the spirit of the Global Virus Network. Thank you so much. So we have some questions which will appear on the Q&A. You have some first congratulations from Ulrike Protzer, and then you have a question. Can you read them, or do you want me to read it for you?
I can read it.
Okay.
I can read it. So one question is, it appears that hyperacetylation is detrimental to the virus, suggesting that NS3 should not be acetylated all the time. So does that mean there is a deacetylase working together with Cat5 gamma? I think that's an excellent question. I can tell you we have not yet identified the deacetylase, but we believe there is one. Our data, especially on the helicase activity, would strongly suggest that it needs really this reversible acetylation and then removing it again, and thereby allowing, the helicase to work. And this is, on our list, and we are attempting that. We believe there is a deacetylase that works in concert, or let's say as a component to Cat5 gamma. Yeah.
Okay.
I think this is what was in the chat.
No, I have, I mean, of course, I'm not an expert, so I cannot, but I mean, the obvious question is always, I mean, you identify some very interesting targets, and you are targeting these host enzymes. What is the possibility, as always, that when you will have really efficient drugs targeting these molecules, actually you will have side effects due to a number of activities in many cells? You see, it's always a concern. This is obvious, but it's always a concern when you have this strategy. So how do you contemplate this challenge?
Yeah, this is, of course, the biggest downside of a host-based, you need a certain window where, it would have hopefully not, many side effects or effects on the host. I think our example, even though again we don't have a drug, targeting Cat5 gamma, it's more like a conceptual advance. For example, it's one specific isoform that may be, really usurped by the virus for its own benefit, which might give us, and this is honestly speculation at this point because we have not really attempted to, in terms of a drug targeting Cat5 gamma. But it could allow, if there's, for example, a specific isoform used by the virus that is not key to, cell function, that could give you a window potentially to target it. So just again, as an example, Cat5 alpha or the other isoforms, clearly you can actually not make, we have attempted to make knockout cells. It's detrimental to the cell. It's also knockout mice. It has been reported they are not viable. Because again, it's a major epigenetic, regulator. But again, the gamma seems to have no cytotoxic effects. What we can see when we, target it. Again, this is using gene targeting or siRNA approaches. And again, this is only conceptually. I completely agree with you. This is, for host-based targets. I believe it would need to be tested for each single target. Again, acetyltransferase kinases, that is, of course, very prominent, targets. But literally for each single target, we would need to test, how can we mitigate any cytotoxic effects, or maybe, what's the window between, the host effects versus the virus. But I think it's a completely valid concern. But I still think we really need to test it from each target, every target at a time. There could be very unique things like specific isoforms, specific localizations. We getting back maybe to the acetyltransferase, the gamma, again, this is all hypothetical at this point, but if we maybe had ER targeting drugs, bringing them only to the ER, whereas, the other, in this case, acetyltransferases or the other isoforms are in the nucleus, that, again, this would allow us maybe to target it, again, at this point, all, hypothetical. But I think for each target, we need very detailed mechanistic insights in kind of also trying being creative, such as, for example, ER targeting, in this case, Cat5 drugs, if we can deliver, it in a certain compartment.
Yeah, no, that's very, very interesting. In which journal has your paper been published this August?
Mm-hmm. In Cell Host and Microbes, so it just came out this month.
Oh, great!
Earlier this month. Yeah.
Do not hesitate to send it to Kevin. You see, we have his GVN list weekly, and also we list the publications which are coming from our GVN centers, and this type of publication might be very interesting. I'm sure that Robert Friedrich, who is now our Chief Content Officer for GVN, might be very interested to connect with you on this.
Okay. Yeah, that would be fantastic.
And also for the flavivirus, the hemorrhagic fever task force. I mean, if you can be part of it, that would be very interesting. So this is something that Alfredo Garzino might, should, and will contact you to, if you agree. I mean, to have you be part, you see, of this task force and watch groups.
Yeah, that would be fantastic.
It would be very interesting. Yeah. Okay, well, thank you so much again, and that was just a start. We are very happy to welcome you as a director of a GVN Center, and we will keep in touch. Thank you so much, and thank you to all the attendees.
Thank you so much, Christian. Thank you, everyone.
Bye.
Bye.